We are now recording, and thank you very much, Jake, for agreeing to give a first Build a Cell seminar of 2021, and it's all yours.
All right, thank you very much for inviting me. My name is Jake Beal. I am one of the leads of the synthetic biology group at BBN Technologies. Technically Raytheon BBN Technologies, but if you think Raytheon, you might get a little bit confused because Raytheon is an enormous, enormous thing that does all sorts of stuff. BBN is a chunk inside of Raytheon that used to be its own thing. You may know us from email was created at BBN, as was a lot of the early work on the internet was BBN, and GenBank comes from BBN as well back in the day. So we're a mostly computer science focused organization that operates a lot like an academic soft money lab. So if you can fund it, you can do it. And there's a whole bunch of us PIs there going off and doing all sorts of crazy things, including synthetic biology for the past decade or so. So I'm really not originally a biologist. I'm originally a computer scientist who happened to be in the right place at the right time during my PhD work to get exposed to synthetic biology kind of taking off. Ron Weiss and I were graduate students in the same group, and Tom Knight was one of my advisors. And so even though I was doing other stuff that was inspired from biology to do computer things, I kept looking back over at the biology and like these interesting things starting to happen, because I wanted to be able to do good, robust, resilient engineering things. And I realized the stuff I was doing over in the computer world turned out to be applicable over back into the bio world where the ideas came from in the first place. That sucked me down into the world of biology. And in order to get the engineering tools I wanted, I've had to engage more and more into fundamental challenges of metrology and data analysis and statistics and protocols and so on, because where we've been in synthetic biology is a world of arts, where to do anything, you have to be a master craftsperson. And it's long and it's hard, and it's not the world of engineering that I want to live in, which I think is very apropos to Build a Cell, where you understand things so thoroughly that you can put things together and predict what's going to happen before you get there. So this talk, I really want to focus on the foundations of what we need in order to get from this world of being a very specialized, slow, delicate, challenging art to routine, predictable, reliable, effective engineering. I want to start here by observing a basic challenge that's going to unite everything I talk about in this talk, which is that engineering doesn't happen in a vacuum. If you are a master painter spending a year in an attic somewhere doing something fantastic with oils, you can work all in isolation. But every engineering field is distinguished by the partition of problems into lots and lots of different specialists who make components, who make modules, who make tools, who make reagents, all these different things that come together to build your system. You don't build a house by going to the forest and cutting down a tree. You buy 2x4s and you buy electrical wiring and you buy shingling, all these things that have been developed for the building of houses. And the way that you know how to put those together is you have to be able to answer a lot of questions about what is this? How do I put it together? What does it do? What should it do? How can I predict that when I put these shingles on my house, it will keep the rain out? So when we want to think about putting things together in bio, we're always going to be thinking, if we're in an engineering world, the same way of I'm going to get the things I use from somebody else, like say this simple prototypical arabinose sensor. We express Ara-C that interacts with arabinose to regulate the PBAD promoter. And here's some data from a flow cytometer showing that it turns on in some particular condition. But to actually put this to use, there's a lot of questions that we've got to be able to ask. Like, on this data here, is 10,000 yellows a lot? There aren't any units on that scale. Is it more or less than 500 blues? Because blues aren't necessarily the same as yellows. Is this curve I'm seeing on the device, is that actually going to achieve the thing I need this device to do? Is it going to give me the signals that I need to drive the other part of the system I want to have respond to 𝗲𝗏𝗄𝗏? What was the conditions under which this data came from? This promoter here doesn't have any label on it. What is it? What's this DNA actually supposed to do if I'm not already an expert who understands 𝗲𝗏𝗄𝗏 inducers? And all sorts of terminology issues too. I'll see somebody write down D+ on their notes about what an experiment had. Did D mean 𝗲𝗏𝗁? Did it mean dextrose? Is it something else? Even things that you might think are the same, like the M9 media. Well, this lab might prepare it a little differently than that lab. And like maybe when they say M9, they actually mean M9 with this little tweak that everybody does because now we know that this is the better way to do M9 for our system, and that just vanishes out of people's lab notebooks. So you can end up with a huge amount of back and forth and pain, and you waste 6 months at a time just trying to figure out what is this thing that somebody's given you. But if we have good units and good tools to help us curate the information about our devices, that can make sharing much, much easier. So this is the idea that I want to return to throughout this talk. If you remember nothing else from this talk, remember this problem. Well, let me I'm going to make the problem incredibly overly simple and say, one thing we could look at is sort of the atomic unit of all engineering. We don't have to understand things perfectly, but we have to have low enough uncertainty that we can be reasonably good chance of getting things right when we want to say, is this thing over here going to satisfy my need over there? Is this nail the right size to pound into the wall to put these two joists together? Is this promoter the right strength to drive this 𝗲𝗏𝗄𝗏 sensor enough that it will respond to 𝗲𝗏𝗄𝗏 to give me as much signal as I need? Can reduce this down, think about it as sort of a very, very basic thought of, I have a thing which has some estimated property value, say the amount of expression that this constitutive promoter gives me. And I have some prediction about what's the acceptable range of that. Do I need 5 molecules per cell? Do I need 100,000 molecules per cell? Some range of values for the one are going to make my system work. But I don't necessarily get to see what those values really are. There's some degree of measurement error in estimating just how strong that constitutive promoter is. There's some degree of model error in estimating what is the range that will actually work. And so we end up with what you can think of as a three-way tension of ways to try to make this better. You can get better at engineering by making your measurements more precise so that you really know that what that constitutive promoter is doing in a very good way. You can make your models better so that you have a better understanding of what you need that promoter to do. And you can make your devices more tolerant so that you don't have to get as precise. So if you have really, really delicate, touchy devices, then you have to be really good at your models and really good at your measurements. The better your devices are, the sloppier you can be with the other stuff and still get something that works well enough. So how do we tackle these things? I see a question here. How many degrees of error would you estimate will be in a cell with 400 genes? It really depends on error with respect to what. This is, I think, one of the really key things in a place where there's a divide between when you put your engineering hat on versus when you put your scientist hat on. When you put your scientist hat on, you want to know what are all the things. But when you put your engineer hat on, you want to know how much do I need to know in order to make a decision. There may be a huge amount that you don't understand, but it may be that if you understand it well enough to make a decision of, is this likely to work? Is this likely to not work? You don't have to understand all the degrees of error. You just have to understand which are the things that pertain to your decision. I'm going to come back to that in 5 slides when I want to talk about how those hundreds and hundreds of genes boil out into more predictable errors in some ways. Right now, though, I want to start by talking about the question of the measurement itself, because this really comes down to one of the really fundamental challenges that we have to deal with, which is that if we don't know how yellows relates to blues, we're completely at sea. This is something that my 3rd grade science teacher would be very embarrassed about, some of the things that we do, because he told me that, you can't just say that, I've got, this is 5. Is it 5 inches? Is it 5 kilometers? Is it 5 grams? We put a lot of stuff out in arbitrary units and relative units, and that's a fundamental strike against reproducibility. I'm going to pick on one of my favorite papers for a minute. This is one of our founding papers in synthetic biology, the toggle switch from Gardner et al. And if you look at it, this really is a beautiful paper. They got something marvelous to happen. But when you dig down a little bit, you'll see there are some things that are a bit problematic and confusing here, which was fine 20 years ago, but we shouldn't still be here 20 years hence. The fluorescence here, there aren't any units, just as GFP. And it's not because it's been turned into molecules, it's because that's coming out of a flow cytometer. And those units are arbitrary units, which happen because of the way that that particular machine happens to be configured on that day of the week, in that phase of the moon, with those particular voltage settings, and so on. So this is something that you can't even tell if it's the same or different if somebody else runs the same circuit. There's also something funny going on if you look at these 2 different sides. Over on the left here, we see this beautiful separation of showing the hysteresis of going up versus going down with the toggle switch, this big gap. Except when we look at the right, this same slice here, you see these distributions are actually overlapping. It's because these statistics are talking about the variation of the means. And here we're seeing the actual variation from cell to cell. It's a lot less separated. The arithmetic statistics over here don't really capture what's going on very well. And so if you try to engineer, thinking that all your cells are either up here or down there, you can get into some real trouble. So what do we do about this? Well, the first thing I want to argue is that it's not actually a biology problem, because almost the first thing that happens whenever I make this assertion that we can do better than this is that somebody will say, but biology is really complex. Cells are incredibly complicated. There's so many genes. We don't understand a lot of them. And somebody will always have a story about, like, there was this one lab and like everything stopped working, and then they found trace selenium had changed in their water supply and like this and that. And like, yeah, biology can be really complicated. But what's happened every time that we've opened up this box and really gone into it and looked at reproducibility is it's turned out that the problems are less about the biology and more about the fact that we've often kind of used that as an excuse to not put enough controls in place on our processes. Here's one example from a study that we ran with iGEM in 2016, which had about 70 labs participating around the world. using plate readers and flow cytometers to measure a set of simple constitutive promoters. What we did in this one was we added in some simple calibrants, just some fluorescein and some suspended silica to calibrate the fluorescence and OD measurements. And what we found is that there's a huge amount of variation from lab to lab in their measurements, but almost all of that was not from anything that was going on in the biology, but from problems that people were having in just mistakes executing the protocols. And we could see those when something went all wacky in one of these very simple calibrants. So if you measured your OD values and it said that water had a higher optical density than silica suspended in water, which is definitely more opaque, then you know that something's gone terribly wrong in that laboratory. Or if you make your titration series of a whole bunch of different levels of fluorescein, and you find that it sort of goes janky up and down and stuff rather than a nice series, you know that something's gone terribly wrong. If your cells aren't growing well, you know that something's gone terribly wrong. There's a whole bunch of places where we can use process controls to go from just a complete Wild West of unusably high variation to nice, tight, only twofold or less standard deviation across nearly 100 labs of undergraduates and high school students. Normalization isn't enough either, because when you normalize to try to, you take your control strain and you say, well, normalize everything to the control strain, that gets you a little way, but it doesn't get you as far as you need to go, because your control strain it can have correlated failures with the experiment strains that you're doing, because the same way that you mess up the protocol will hit them both. Having independent calibrants helps a lot. Also, getting your units and statistics right turns out to be very important. So normalization, didn't get us as far down as just pruning the stuff where something was clearly gone terribly wrong in the protocol, as showed by the independent calibrants. So when you get this right, we can get standard units for fluorescence and OD in plate readers, and also standard units for fluorescence and for size in flow cytometers, where those units even are related to molecules and match between flow cytometers and and plate readers. So you can get the exact same units. They're related to a real physical quantity because they're calibrated to equivalent fluorescence of fluorescein or Texas red, another dye like that. And we've been able to validate that these things work in the hands of hundreds and hundreds of labs over multiple years. So we don't ever have to put up with an arbitrary unit for fluorescence ever again. Other people have done similar things for calibration of RNA measurements for RNA-seq. And it can be extended out potentially to other types of measurements as well. So I think we really need to stop accepting arbitrary and relative units because they really put us just out to sea in terms of our ability to reproduce and in terms of our ability to interpret and design with what's done. Now, once we've got good measurements, we're not quite where we need to be yet, because we also need to have some idea of what the error on those measurements is like. And this is where I want to draw your attention to a phenomenon that you see a lot if you work with single cell measurements like in flow cytometry, which is that you see a lot of distributions which look like nice, balanced, sort of Gaussian-like distributions on a log scale. These here, for example, are taken from some of 𝗀𝗆𝗄𝗆 devices for cello. And why does it have this shape? Why does it have this structure? Well, some people think that this might be because of stochastic noise. Turns out that's not a sufficient explanation, because that should mean that these get narrower as they go up when they stay about the same size. It turns out there's a really good null hypothesis for why you'd expect to see geometric statistics in a really complicated system like a cell with hundreds of genes that may be affecting the transcription and translation processes. And that's because there are a huge number of things that we are not attempting to control that have to do with transcription and translation. All the different elements that come together to form and chaperone the transcription and translation processes, the supplies of the nucleotides and the amino acids, the tRNAs, the energy, the cell state, all this stuff has impacts on it. And you can actually model it all as complex catalytic reactions, which are essentially varying the reaction rate of the basic transcription and translation processes, which means you can model it as multiplying a whole bunch of rates together. And just like when you flip a bunch of coins and add them together, the central limit theorem in mathematics says that that should give you a Gaussian. When you multiply a bunch of rates together, that same central limit theorem says you should expect to see something that's Gaussian on the log scale, a log normal distribution. So that's the baseline assumption that you generally ought to think about whenever you're thinking about gene expression. And that implies that you should use geometric statistics rather than our sort of normal arithmetic statistics in your analysis. I see a question: does robustness of the cell affect the distribution? Absolutely. And if you have some single factor that dominates, you can end up with other sorts of things. But your baseline, you should always think about your baseline being a whole bunch of little factors that give you a log normal, which then may get modulated by big things hitting your cell, like say some of the cells, being killed by an antibiotic, while others are not, because they either have or haven't mutated out, one of the pieces of the system. So there's, but your baseline is a distribution like this, and it may be wider or thinner based on the heterogeneity of your population. So with these foundations of good units and good statistics for interpreting them, we can then come back to our sort of base challenge and move out into making better models and better devices. There's a lot of work that I have been engaged in these areas. Other people have as well, for predicting cascades and other composite systems, or engineering to make promoters that are much better insulated so that to their genetic context, so we can shuffle around a circuit and have the same behavior in all the situations, or high performance devices once we have the right statistics to know what high performance even means. Turns out fold isn't enough. So all right, so we've got a bunch of stuff that we've made progress on. We can get some things that are worth engineering with, but we haven't yet solved the whole sharing problem. And I want to point here to, again, so far we've just dealt with this upper right bubble here, of is 10,000 yellows a lot or not? And in fact, one of the things that I want to point out, we are missing a lot of implicit information that's typically in a lab notebook, in a discussion between postdocs or graduate students, even down to, in this system with this arabinose detector, where's the arabinose? You have to have a human expert who somehow knows that 𝗲𝗏𝐶 and 𝗃_BAD have something to do with arabinose in the typical GenBank representations that get tossed around. So one of the key things for being able to encode this information in a way that we can package it up and be able to share it effectively in a way that machines can help us catalog and share and check that enough information is there. So we need to be able to represent the whole system. That's where 𝗙𝗏𝗄, the synthetic biology open language, is, I believe, one of the pieces of that particular puzzle. This is a community, international community of folks across academia, industry, government, who all have these same problems of information sharing and have been essentially constructing a representation. It's now in use fairly widely within certain communities for representing the whole design, build, test, learn cycle. Not just the genetic construct, not just the models, not just the data, but also the connections between them and how they connect out to other data sources, combinatorial designs, both the structure and the function, etc. Once we have something that we can represent all the things we want to talk about, we've still got a problem of actually getting things from the lab notebook into the computer. This is a big challenge. I think of it as the curation challenge, where your typical process right now is that you've got a genetic design that's been made in something like benchling or Excel or what. You've got a description of your experiment, which has probably been written up in Word or some electronic lab notebook software, or maybe just a physical paperback lab notebook still. And a human goes off to the lab and they do the protocol. Maybe you get some automation if you're lucky to help it be more high throughput, maybe not. Get a bunch of data files and you get a bunch of, well, probably spreadsheets out at the end. What's the relationship between these things? It exists only in the head of a human being, and that's going to be very hard to transfer somewhere else. Right now, typically, trying to connect these things together is a post facto process where it's like, oh, we want to share the protocol. Okay, we got to go dig back. We got to find out this all. We're going to publish. We got to get just enough so that it's not going to be painful for the reviewers. But, you're dealing with experiments that happened 18 months ago, and, that postdoc left and went to another lab, and a huge amount of information falls through the cracks here. Also, the people who are experts at the bench probably aren't experts in things like 𝗙𝗏𝗄, and they shouldn't have to be any more than they should be experts in database design in order to use Excel or benchling. So there's a bunch of work that's been going on with the DARPA SD2 program to leverage 𝗙𝗏𝗄 and a data store called SynBioHub to link together all these different pieces. And I'm not going to talk about all the bits and pieces here, but to basically have a little bit of machine assistance to let you do all this curation incrementally and proactively rather than post facto. So for example, we've got a system called Syndict. This stands for the Synthetic Biology Curation Tool, that you feed it a design in GenBank, and it will go through, annotate it, and try to figure out what this is supposed to do. It's in specific, right now, it's configured just to think about genetic circuits. So, regulatory networks. It tries to extract a genetic regulatory network based on what it knows about repressors and activators and inducers and so on. And it'll tell you, I think I found your circuit. Here's what I think it looks like. At which point, that's a really great place to find errors in your annotations, find errors in your encodings, sometimes even find errors in your designs. Some of the systems we've applied this to have turned out to have some really unexpected things in there that turned out the system was malfunctioning, because the system was not actually the system that the person thought that they had built. But a computer is very stupid. And a computer will happily show you, well, here's, I didn't know how to fill in the gaps, boss. Here's this design and I don't think it's going to work. And can you help me? And then it's up to you to see. But if you've done it well, then the computer can figure it out. There's a similar thing with the intent parser. Is a system that we've got set up that looks at Google Docs, where if you write your experiment plan up in a Google Doc and use just a little bit of standardization in how you set up tables to describe the combinations of conditions and strains and constructs that you want to put together. It provides you templates to help with that. If you organize yourself just a little bit, then it can automatically extract out plans, which both we can do checking on, and we can send into automation with partners like Stratios, formerly Transcriptic, to be able to run at high throughput. There's a whole bunch of different pieces here that all bit by bit help push the process of explaining what you've done upstream to the experiment design stage in small, low pain chunks, rather than downstream at the end of the process when you just want to be done with this or where you're having to revisit it far later. So this can potentially make it much easier to capture all the knowledge about the systems that we're doing in a way that's friendly to the people at the bench and not just computer scientists. So with all these pieces, I think we're at the point where with these and other similar tools that people are working on can start pulling together the knowledge in a way that can enable much better predictive engineering, where we can actually answer all of these questions and actually put arabinose in our arabinose detector description to build up from these basic foundations of metrology, good representation, strong devices, better models, to be able to answer the questions we need to do effective engineering. What's going to work? Why isn't it working? Can I even do this with the parts I've got? What sort of systems are reasonable? To be able to build the more complicated systems that will let us attack the wide range of applications where synthetic biology can potentially transform our world. And with that, I'm just going to leave us on an acknowledgement slide talking about a bunch of the organizations that have been involved. And I see another question in the chat that I'm going to answer. Is 𝗀𝗏𝗏 platform agnostic? Can you encode genetic pathways with all the information about the expression system? Yes, there's no assumption about any particular chassis or so on. 𝗀𝗏𝗏 is platform agnostic in the same way that something like GenBank or FASTA is platform agnostic, except it's a much, much richer language. And people have been using it for mammalian systems, for yeast, for plants, for metabolic engineering, for circuit engineering, etc. See another question. Can we build a framework for designing the most common synthetic cell types to share with the community? So I think that's a great question. I personally don't know enough about synthetic cells to know what's in common and what's not for you. My experience working with standards organizations like 𝗀𝗏𝗏 and loose collaborations like iGEM is that it's often people often want to try and get everything right. And then you get wound around this big challenge of we have to get all the things right at once. And that's often not the most productive way to go forward in terms of making things coherent as a community. I found it's often much better to start with these are the things that we all definitely agree on. Here's our sort of lowest common denominator. We're going to use units for fluorescence. And then from those, you can build up into the more and more sophisticated things. To do that, you need to make sure that the system you're using to share with is open so that you can have custom information in as well. 𝗀𝗏𝗊 is an example of a system that does that, not because it's special to 𝗀𝗏𝗊, because 𝗀𝗏𝗊 is built on a World Wide Web consortium standard called RDF that is really good at open knowledge. It's the same thing that people build bio-ontologies with. So I think that there could be some really constructive discussions about what are some basic synthetic cell types, what are the things that you want to standardize around them, but don't go too far. Start with the lowest bottom pieces and just try and get those right, and that'll support a lot of things moving outward. I see a question from Anna Marie. Thank you, glad you appreciated the talk. What's the biggest source of error in designing genetic circuits? Experiment or pipetting error, some intrinsic error sources? What I have observed so far is that I'm going to modify the word from error a little bit because I want to talk about barriers instead. So the first biggest barrier that I see is that a lot of times things just aren't even comparable because there's no standard to compare them against. There are no units. So like, an example of this, 𝗀𝗏𝗃 and his lab have done a lot of great work with designing some really high performance devices in the 𝗀𝗆 system. However, those devices have all relative units. So you have to reference them to a particular plasmid that they built. So there's no way to really integrate them with anything else outside of their system. It's hard to know what, are they high, are they low? What does 10 to the 2 mean in there? So just getting through that is the first biggest issue. That typically then, once you know what you're dealing with in terms of some reference to some sort of physical standard, now the biggest thing that I see as a problem is often leakiness, and that you have a circuit that is just not performing well enough that you could ever build something useful out of it. And often that's obscured by the way in which things are reported in our publications, so that you have something where it's working in the sense that the mean of this was 5-fold greater than the mean of that. But if you plug it into something else, you just don't have enough signal there. So another really important metric that's missing a lot of the time right now is signal to noise ratio, SNR. This is a measurement from the information community, which is universally applicable. It doesn't matter what you're carrying information on, whether it's wireless radio or electronics in your computer or biology or tapping sticks on a wall. It's mathematical universal. It just says, here are two signals. How easy is it to tell the two apart from one another? And we need to start using this more in our characterization of the systems we're working with to ask, how well can I tell this condition of my system from that condition of my system? It's like a t-test, but much stronger. From an engineer's perspective, anything that you have to use a p-value to distinguish is probably not useful for engineering. You need much, much stronger separation than that. So, and then there is a huge amount of error that comes through, like in experiment protocol. And that's, again, it's error that's completely invisible unless you have a basic standard to reference things against. So we don't even know what goes wrong in a lot of labs or which things have or haven't gone wrong. Moving to the next one, a question from Tim. Seen knowledge capture systems that try to get experimental metadata in a standard format by asking the scientists to enter all their data in a standardized form and workflow. So the big problem that I've seen with things like that is that they're often overly restrictive. The reason why we went to trying to, we're not actually parsing unstructured documents. What I see as the middle road here is what I think of as the adventure game form. I may be showing my age here, but when when I was a kid, there's these text adventure games on computers where you'd say like, go north, or look wall, and you could use some very primitive English to talk to them. And as long as you and the computer sort of talked similar language, you could go a really long way. It turns out parsing unstructured text is really hard. Nudging people to talk in a structure that machines can understand is actually pretty easy. So that's the middle road that we're trying to do, where there's a lot of flexibility, but there's also enough nudging so that you can tell if the machine can understand you or not. And so far, at least, this is something we're doing in the DARPA SD2 program. The lab folks have responded really well to that. And it's proven flexible enough that even when they come up with a very different type of experiment, we're not trying to go deep enough that we get into trouble there. Let's see, is there a source of what in hardware would be jelly bean parts, like 𝗙𝗏 definitions of arabinose to refer to, so everyone's using the same term? It's funny that you should ask about the same term. That's actually one of the other issues that we deal with in this curation challenge. This system called the Project Dictionary came from the observation that we couldn't get people to agree on the same term, and it seemed to be a fruitless effort because there are multiple different terms to refer to something. But if you, so this dictionary lets you do post facto integration of different vocabularies that different people and also different lab automation systems have for the same thing. So that era in one lab means arabinose, in another lab means A+, in a third lab, and you can refer it to a grounding definition that says, actually, everybody's talking about L-arabinose, and here's its 𝗀𝗵 ontology pointer. So a really important idea of this is that although we often use different words, usually if you ground out in a fixed definition, like some of these great databases out there, like 𝗀𝗵, UniProt, like a GenBank or 𝗙𝗏 specification, you can get something that everybody can agree what that is, and the labels for it are emergent instead. And yes, 𝗙𝗏 has its ontology. We make very heavy use of existing ontologies as well. 𝗙𝗏 wants to integrate, it doesn't want to reinvent. Question from Derek, does 𝗙𝗏 include membrane and architecture? So there is the ability to talk about that if you want. I don't think people have done much exercising of it. But the things that I know about how in 𝗙𝗏 you can talk about, this plasmid is contained within this compartment that's contained within this strain that's contained within this media sample, and you can attach biophysical measures to any of those. I think you should be able to do the same thing to the membrane, where if you talk about this cell. So there's a set of topological and geometric relations in there that I think would give you what you're looking for, Derek. Let's see. Hagen says, is it possible that a softer approach might be more productive in synthetic biology? For example, you wouldn't subject a musician or a ball player to such rigor and precision. Hagen, I'm afraid I have to take issue with that notion. Musicians are subject to an incredible degree of rigor. My 8-year-old daughter who plays the guitar has a tuner. This is a little thing that her guitar teacher, wants her to, pluck the G string, and this little device will say, it goes up to the red because the G is a bit sharp. and it goes down to the red on the other side when G is a bit flat. Same thing, a baseball player, there is absolutely a very clear measurement that pitcher is getting, clocked on how fast they throw the ball every time. I think that, these types of things, this is the sort of place where you've got metrology. It's not what you do with the art, but knowing that that your G string is producing a G, that's fundamental. And I think that that's the level that we should be asking our biologists to achieve as well. If I put in a promoter and it's a constitutive promoter that's supposed to produce, 10,000 methyl, in a standard configuration, then if it's producing 100, I know that something is terribly wrong with my cells. If it's producing, 10,758, then I know it's close enough. I can put tolerances on there. How flat do I allow my notes to be? Let's see, Lars says, this question might be a bit off topic, but you mentioned iGEM. Would it be possible to talk? Well, I think that question may be for Kate rather than me about giving a presentation about an iGEM team. So I'm gonna
I feel called out, but I'm reading this right now. Lars, shoot me an email. We are trying to develop something like a coordinated synthetic cell subgroup within all the iGEM teams that are interested in this area, but nothing happened so far. So, and if you want to give a presentation about it, please do. Shoot me an email and we can talk about it.
All right, coming to the next, Jess says, you mentioned the need for standards to compare in absolute units. I'll say, by the way, if I said absolute, my colleagues at NIST would tweak me on a little bit. I've calibrated units calibrated to an independent calibrant. It was what I really mean, because we can't necessarily get everything exactly down to molecules in an efficient way or things like that. The question, what would be the mechanism to develop standards like that for our community, where we're an international crowd trying to build a whole cell? So the way I always think about this is that ultimately measurement exists in a context. Every measurement is actually an act of communication with something that's going to consume that measurement. So the key question here is, what is it that you actually need to know to determine whether something is being successful or not? In engineering, genetic regulatory systems, a lot of stuff really boils down to a relatively small number of metrics that turn out to be very key, because the engineering decision that we're typically making is what promoter do we connect to what payload. So we need to know things about that basically talk about what promoters do with respect to payloads. I don't know what your metrics are for the cell, but I'd start thinking about that basic question of, if you did something cool with a cell and I want to use it, what's the typical point of engineering modularity where you can take something from one context and try to use it in a different context? From something up above, it sounds like membranes might be an important thing to talk about. And so if you have some standard way to talk about membranes, because if one of the things you can do is swap around membrane properties, but it's the decisions that you make that you want to be guiding. Every measurement should speak to a decision, because I can measure so many things that turn out not to be relevant. Sometime an interesting exercise to do is look at a set of blueprints from something, whether it's, Google up blueprints, find blueprints for a house, for a mechanical device, for something, and look at all of the things they didn't measure. You'll see that there's lots and lots of numbers for measurements. Like if you made a plan of my living room, there'd be measurements for the ceiling, measurements for the windows, things like that. But nobody would measure the distance from the edge of the couch to a window on the opposite corner of the room. Most of the measurements you could make, you don't. And if you think about which of the measurements you don't need, that can help guide you. Let's see, I like Emma's joke about artificial organisms tapping sticks. Wandering down there. So M9 was mentioned. So in some repository, it could refer to, say, this M9 like a DOI, but for 𝗀𝗏 entities. That is exactly right. One of the typical ways to do this is, so for things that are well-defined somewhere else, we don't redefine it in 𝗀𝗏, you can just point to it. So I don't know if anybody's made a catalog of media recipes, but you might be able to, like if there's a particular media recipe on protocols.io, you could point to that. You can actually just use a DOI right there, and 𝗀𝗏 will actually let you do external references of that type. So you can say, my media is an externally defined object and it is over there on protocols.io. You can also define it within 𝗀𝗏, and you can put that, there are repositories that are out there like synbiohub.org, and you can set up your own repository too. And then you can say this M9 with a URL that refers to it, and that URL tells you exactly what you're talking about, because there's where the machine readable definition is. That's what we do in SD2. That's what we do in a number of other places as well. iGEM has that. There's an 𝗀𝗏 iGEM repository too. That's a mirror of the core parts repository. Let's see, and going up through the talk here. A follow-up regarding external references: is there guidance to prevent link rot? So the bioinformatics community actually has a really fantastic resource to help with this, which is a system called identifiers.org. So identifiers.org is a archival way of linking to entities in places like 𝗀𝗏𝗵, UniProt, any ontology that you're using, etc. So anything that's supposed to be a fixed resource like that, you can use identifiers.org to get persistent links. And one of the values of those is that they track if that entity gets renamed. So like if UniProt changes how they do their referencing, they push that back to identifiers.org so that your references will remain the same. That doesn't necessarily help with your designs, but you can ground a design. If you just have like a genetic sequence, well, if it's in GenBank, you can link to it there. And there are attempts to make repositories that are going to be long-term for 𝗀𝗏 as well. So JBay has their ICE repository is an 𝗀𝗏 enabled repository. There's this synbiohub.org repository. Those are both not necessarily guaranteed to be archival on a span of decades, because they're tied to particular investigators and organizations that haven't made an archival commitment yet. But I think that that's something that's readily doable by organizations in the same way. The technology is there. All you need is people to commit that they're gonna, that they're gonna use particular things that do have persistent storage. So I've come to the end of the questions that have showed up in chat. Let me pause and take a breath here and see, are there other things people wanna ask or bring up or discuss?
I want to say thank you very much, Jake. That was really informative, and that points out how many things our community has to think about to make it work.
Thank you. And my goal at the end of the day, I don't want everybody to be thinking, oh my God, I have to think about so many things. I want everybody to be thinking, I can grab these tools that let me not think about those things. So some of the tools that I referenced are all free and open online. So the calibration tools for flow cytometry, our 𝗙𝗵𝗶 flow analytics package is on GitHub. iGEM has a set of free and open tools for plate reader calibration. There's all the 𝗀𝗏𝗏 stuff is free and open online. All these SD2 tools that I was talking about, they're up on GitHub, free and open. So I want to encourage people to engage with these things, try to make use of the tools, and bring your use cases to these communities. One of the challenges we often have in the tooling communities is that folks from the lab communities are sometimes not aware, or they'll try it and these things are beta. They haven't necessarily been tried on your particular use case before. You hit bumps and then you say, oh, I guess I can't use that tool. The biggest thing you can do to contribute to making the tooling world that you want to live in is when you hit a bump, rather than walking away from the tool, file an issue and talk to the maintainers of the tool, because often they're really eager to have users that they can support with their use cases. So I encourage you, everybody at the bench, think about a tool that you'd like to try to use, and not if, but when you run into problems with that tool, ask the maintainers for help, because they'll want to help you. Thank you.
Thank you very much, Jake, and we'll post your talk online, so it's going to add to those resources.
All right. Thank you much.
Thanks, and bye, everyone. Bye.